part of raising crops is improving the genetics of the crops we plant. Although this section of the course is largely about genetically modified organisms, what we commonly refer to by the acronym GMOs, one cannot understand genetic modification without understanding other breeding techniques, including induced mutations and traditional selective breeding. The purpose of this video is to make sure we understand how the genes of crops are altered using these tools. We will start at the level of the crop's DNA and the genes it contains, and articulate how an organism's genes are passed down to the offspring in traditional sexual reproduction. We have already learned about DNA, including the nucleotides it is comprised of, and the massive amount of information the DNA contains. Here, I'm going to use a very simplified version of inheritance. Remember that the DNA contains information, instructions on how an organism should be built. So I'm going to represent a very small strand of DNA as containing only 3 sets of information. Let's suppose the DNA contains information on only 3 attributes of a plant: the size of the seeds it produces, its vulnerability to drought, and its resistance to a pesticide. I call it the mother of the subsequent offspring, even though a plant may have both male and female parts. Each symbol refers to a different gene. Remember that the mother's DNA comes from her parents. She gets 1 strand of DNA from her mother and 1 strand from her father. The attributes of the plants are then determined by the DNA from her mother and her father. Sometimes one gene is dominant over another, and it solely determines the plant's attributes. For example, in this scenario, her father's gene for seed size is dominant over her mother's, so only her father's gene determines the size of her own seeds. If this gene codes for large seeds, she will have large seeds, regardless of her mother's gene for seed size. In regards to vulnerability to drought, her mother's gene is dominant, and so her ability to withstand drought is determined solely by her mother's genes. Other times, there's no single dominant gene, and both genes simultaneously determine a plant's attribute. Here, the plant's ability to resist a pesticide, that is, not be harmed by the pesticide, is determined by the genes passed down to her from her mother and her father. And so, our simplified DNA has instructions for only 3 different attributes, and there are 2 genes for each attribute, 1 given to her from her father, 1 given to her from her mother. Now let's suppose this mother is bred with a male to produce an offspring. The father also has 2 genes for each attribute, 1 given to him by his father, 1 given to him by his mother. What I want us to do is to think about how the genes from the mother and the father are passed down to their offspring. Although the mother has 2 genes for seed size, she can only pass down 1 of them. It could be that the mother passes down the entire strand of DNA given to her by her mother, or the entire strand given to her by her father. If it were true that each parent could only donate 1 of her 2 complete strands, then because each parent has 2 complete strands of DNA, 2 parents could only give birth to 1 of 4 types of individuals. That is, if they had more than 4 children, at least 2 of those children would have to be identical twins. But of course, we know this is not the case. Organisms, including humans, can have much more than 4 children, all of whose DNA are not identical. What really happens when a parent donates 1/2 of its DNA to its child is that it shuffles its 2 strands of DNA before it passes a strand of DNA to its child. They can give to its child a hybrid DNA strand made of elements of DNA both from her mother's mother and the mother's father. To illustrate, when it comes to determining which of her 2 genes corresponding to seed size will be passed down to her child, the reproductive process randomly picks from 1 of the 2 genes. The same goes for the genes for vulnerability to drought and to resist the pesticide. This means that the offspring has a 50% chance between acquiring its grandmother's gene for seed size and its grandfather's gene for seed size. Notice that although the two strands of DNA are shuffled before bestowing down to an offspring, each gene always stays in its proper location in the DNA. That is, the top part of the DNA, corresponding to seed size, is never moved down to the middle or lower genes, referring to drought or pesticide tolerance. Because of this randomization in determining which genes to pass on to offspring, and because the DNA is so large, much larger than the 3-gene DNA model we are using here, there is almost an infinite number of genetically unique offspring two parents can create. Suppose we now wanted to have an organism with the following DNA. This organism has a green gene that looks like a file cabinet in the space corresponding to the gene vulnerability for drought. Perhaps we believe a plant with this unique gene would better handle droughts than other plants of this species. Let's call a plant with this green gene in the middle space a drought-tolerant crop. Notice that this gene is not present in the middle portion of the DNA for either the mother or the father. So with the mother and father bred and biology proceeded as usual, it would be impossible for them to create a drought-tolerant crop. So if we really wanted this drought-tolerant crop, we must introduce this gene somehow. One way is to find a new mother and father who do have this green gene. This is an ancient method of creating new plants and animals. But today we are doing this in a very scientific manner. We first identify the gene that bestows a plant with drought tolerance. Then we sequence the DNA of the various plants to determine which one of those has this drought-tolerant gene. This is called molecular marking. Then we deliberately breed those plants known to have that gene. So traditional plant breeding today uses the most advanced technologies in genetics. Another option is to rely on the fact that genetic mutations will happen, and given enough time, when passing along their genes, some of their genes will randomly mutate into the green gene that gives us a drought-tolerant plant. The problem is that genetic mutations happen only rarely, and we're not willing to wait thousands of years for the right mutation to occur, especially when we can speed up the rate of genetic mutation using chemicals and radiation. Consider the Clearfield varieties of wheat in Oklahoma. One of these varieties is shown here. These varieties were created by using chemicals to speed up the rate of genetic mutations in wheat. And some of those mutations bestowed the plant with the gene that makes it plant resistant to certain herbicides. This allows the farmer to spray herbicides right on top of the wheat, and though the herbicide will kill most of the weeds in the wheat field, it will not harm the wheat itself. To see how valuable this mutation is, compare the Clearfield variety on the left to another wheat variety on the right. The other wheat obviously has much more weeds, which reduces the amount of wheat the farmer can harvest. These Clearfield wheat varieties could also have been made using radiation to speed up the rate of genetic mutations. Just as OSU created a new variety of wheat by encouraging genetic mutations, one could do the same thing to acquire the drought-resistant plant with the green gene. Now remember, this Clearfield variety of wheat was created by using chemicals to encourage genetic mutations in the wheat. This is not an example of genetic modification, though. That is, the Clearfield variety is not a GMO. Now you might ask, why? To answer that, let us now discuss what genetic modification is. One way to get that green gene in the middle portion of the DNA is to simply cut the green gene out of a different part of the plant's DNA, perhaps the lower part if the green gene is there, and then insert it into the middle portion. Or cut the green gene out of a similar plant and insert it into the middle part of the DNA. Or cut the green gene out of a completely different organism, perhaps a bacterium, and insert it into the middle part of the DNA. Any and all of these methods are examples of genetic modification, and the resulting plant would be considered a genetically modified organism, GMO. If the gene was cut out of the same plant species or a similar species, the resulting plant would be referred to as a cisgenic GMO. If the gene was cut out of an entirely different organism, the resulting plant would be called a transgenic GMO. Most GMOs in agriculture are plants, and most are transgenic plants. Consider an example of how genetic modification can improve and protect our food. The vast majority of the bananas we eat are of the Cavendish variety, and they are not bred naturally but are propagated by cloning. This means that any disease that threatens one banana plant threatens all banana plants because they have virtually identical DNA. One disease is called the black sigatoka fungus. However, scientists are trying to develop bananas resistant to black sigatoka by using radiation to encourage genetic mutation in bananas. The tropical race 4 disease is also a fungus, and it's being confronted using genetic modification. Defenders of GMOs will often remark that genetic modification is nothing new. We've been genetically modifying plants since the beginning of agriculture. Simply by deliberately choosing to plant seeds from crops that produce the most food, we are modifying the genes of the plants that we cultivate. Because we prefer corn plants that yield more rather than less food, we have transformed an ancient grass called teosinte, on your far left, to a plant that yields more and bigger seeds, ultimately resulting in a modern corn plant on your far right. So no, genetic modification is nothing new and is the normal part of agriculture. But it should also be added that genetic modification is a much more precise and reliable tool for acquiring better crops. I've never understood why the public dislikes genetic modification but raises no protest against inducing genetic mutations in plants through radiation and chemicals. However, we should also add that genetic modification allows us to alter genes at a much faster pace and in more diverse ways than traditional plant breeding or radiation and chemical-induced mutations. For those who do not trust the corporations doing the genetic modifications, the last thing they want is for the corporations to have more flexibility and more speed in creating new plant varieties. And therein lies the modern debate about GMOs.